Okay, guys, time to understand DNA technology. Okay, so basically, everybody's DNA is different. Except for identical twins, no two people have are genetically alike. But that variation is only 10% of your total DNA. So most of the DNA is basically the same in all humans. But that 10% helps scientists to identify whose DNA it is, which is pretty cool. So 98% of our genetic makeup does not code for any protein, and that is called non-coding DNA. So non-coding DNA contains many length polymorphisms, or variations in the length of the DNA molecule between known genes. So in a DNA molecule, you have genes, right? So between those genes is differences or variations in the length of the DNA molecule. So those are called length polymorphisms. So it basically changes the length of the DNA molecule between the known genes. And those variations that changes the length is known as length polymorphisms. And that's contained in a lot of non-coding DNA. So some length polymorphisms in non-coding regions come from short repeating sequences of DNA. And an example of that is a repeating nucleotide sequence that might be CA, or adenine, cytosine, I believe. Okay, double check. Yep, cytosine and adenine. Not sure how to pronounce it. But anyway, so that is an example of a repeating nucleotide sequence that is short. So these sequences can repeat a few or many times in a tandem. A tandem is one behind another sequence. So basically it's like, CA, and then it's like one behind another, and another small sequence will be right after that. So it's like repeating. And those are called variable number tandem repeats because it varies. Variable number, there can be varying number of the tandem repeats. So the number of tandem repeats at specific places loci in DNA varies among individuals. So the tandem repeats at specific places is different among different people. So those spaces between the repeats is different, and that's one of the things that makes people different. For each of the many VNTR loci in a person's DNA, he or she will have a certain number of repeats. Geneticists have calculated how frequently VNTR used in DNA identification occur in the general population. So geneticists have calculated how frequently it's used. And with these numbers, they can determine how rare a particular DNA profile is. So that is DNA identification. So now it's steps in DNA identification. So the main steps that are involved are isolating the DNA sample and making copies if it's needed. Then it's to cut the DNA into shorter fragments that contain known VNTR areas, and to sort the DNA by size. And number 4 is to compare the size fragments in the unknown sample of DNA to those of known samples of DNA, and that's basically how you identify the DNA. And if a match occurs between the unknown sample and a known sample, then a person's identity can be confirmed. So basically, if there was a robber and there's a suspect, they would be able to match, or they would be able to see whether the DNA from the crime scene matches the one of the suspect. And if it does, then it's that same person. So the first step is copying DNA, and you use polymerase chain reaction. So basically, the DNA that you find from a crime scene or from human tissues is probably present in very small amounts. So you have a very small amount of the DNA. And in these cases, scientists need to make copies in order to have enough DNA to use for DNA identification. So polymerase chain reaction, PCR, is a technique that quickly produces many copies of a DNA fragment. So basically, through this process, you're able to create copies of it. So in order to do that, what you do is First, the PCR requires a template or a DNA fragment that contains the sequence that the scientist wants to copy. And PCR also requires a supply of the 4 DNA nucleotides, heat-tolerant DNA polymerase, and primers. So what are primers? Primers are artificially made pieces of single-stranded DNA that are 20 to 30 nucleotides long and must be present for DNA polymerase to initiate replication. And primers are complementary to the ends of the DNA fragment that is to be copied. So basically, primers are something that scientists make artificially in order to be able to copy the DNA. So basically what happens in PCR is you add DNA polymerase, free nucleotides, and primers and heat. So the DNA strands separate, and then they cool, and they allow the primers, which are the artificially made pieces of DNA, artificially made pieces of single-stranded DNA. And so those primers like kind of go and match with the DNA strands that are separated. And then the DNA polymerase that was part of the equation then adds the free nucleotides to complete the strands. And then each double-stranded DNA is identical to the original DNA. So basically here, these two strands, they separate. When you heat it, they separate. Then you allow it to cool while adding primers. So then those primers bind with the single DNA strands. And then the DNA polymerase, which synthesizes, I should say, DNA, naturally, would be able to add those free nucleotides that we also added to create new DNA strands. And you heat again to repeat the process. So that is PCR, polymerase chain reaction. So the second step is to cut the DNA using restriction enzymes. So to cut long DNA molecules into shorter pieces, biologists use bacterial proteins called restriction enzymes. So restriction enzymes recognize specific short DNA sequences and cut the DNA in or near the sequence, as figure 13-2 shows. Some restriction enzymes leave DNA overhangs that function like sticky ends so that other pieces of DNA with complementary sequences can bind to them. So what that basically means is, so here's your DNA, your vector DNA, where you're trying to test. So then restriction enzymes basically break the DNA into like these two just separate parts. As you can see, these two would have gone together, but it didn't cut in a straight line, but it kind of cut like that, like jaggedy. So this is the sticky end. So those sticky ends would bind with this. This is a DNA fragment of interest. So what is a DNA fragment of interest? I'm not exactly sure about that, but it's probably something from some DNA from a separate place. Let's read what it says. Figure 13-2, the restriction enzyme in this figure recognizes the sequence GAA, guanine, adenine, thymine, cytosine on each DNA and cuts each chain between the G and the A nucleotides. I see. The G and the A, the G and the A. And DNA fragments with sticky ends result. So basically it was cut there. So then the DNA fragments of interest are probably strands of DNA from another sample or that were artificially made. So basically those sticky ends would attach here. You can see they're kind of copies of what was cut. This part is basically here and this part is basically here to attach. And that is recombinant DNA. So basically to make more of the DNA. So now it's sorting DNA by size, which is the third step. So DNA fragments can be studied using a technique called gel electrophoresis. So gel electrophoresis, I don't know how to pronounce it, separates nucleic acids or proteins according to their size and charge, as shown in step 1 of figure 13-3. So here we can basically kind of see the process. So Step 1 of that shows that the DNA samples are cut with a restriction enzyme, like what happened there. The DNA is then placed in wells made of a thick gel, as shown in step 2. So the DNA is cut with those restriction enzymes, so this process occurs. Then they are placed in wells made of a thick gel. So this is a thick gel. So the restriction fragments, thick gel, an electric current runs through the gel for a given period of time. Negatively charged DNA fragments migrate toward the positively charged end of the gel. Smaller DNA fragments migrate faster and farther than longer fragments, and this separates the fragments by size. So, an electric current is run through this gel, and the negatively charged DNA fragments migrate toward the positive end, and smaller DNA fragments migrate faster and farther than longer fragments. This separates the fragments by size. So, as you add charge, basically what happens is the smaller and shorter the fragment is, it goes farther and it goes faster, while the longer fragments of DNA go slower and they don't go as far, so they stay kind of where you introduced it into or placed it into this wall, while the shorter fragments move farther. And this basically separates the fragments by size. In step 3, the DNA is transferred to a nylon membrane and radioactive probes are added. The probes bind to complementary DNA. So here is the transfer. So this is a nylon membrane. So you add a radioactive probe that binds to the complementary DNA. And an X-ray film is exposed to the radio-labeled membrane. The resulting pattern of bands is called a DNA fingerprint. So the radioactive probe is placed, and then that probe binds to complementary DNA. And an X-ray film, when it's exposed, since it's radiation, to the radio-labeled, since after you place the radioactive probe and it binds with the complementary DNA, those are radio-labeled, so you would be able to see the radiation. The radio-labeled membrane would be exposed through an X-ray film, so this is a completed DNA fingerprint. And those are the resulting pattern from the different sizes of DNA fragments, and that is sorting DNA by size, which is called gel electrophoresis. I don't know how to pronounce it. A DNA fingerprint. DNA samples are cut, transferred into a nylon membrane, exposed to radioactive probes. Exposing radio X-ray film to the membrane makes the fingerprint visible.